Yes. Thank you very much. So now we have Dr. Gianni Pertici from EB Switzerland, and he will give the point of view of the company, no?
Yep.
For this translational and clinical approach of biomaterials. Thank you, Gianni.
Thank you, Serena. So thank you to everybody. It's a great honor to be here today after this pandemic situation. So just a few words about me. I'm a chemical engineer. I did my PhD in biomaterials. I arrived in Switzerland around 12 years ago as head of a research lab in Swiss Stem Cell Bank. Then I moved to University of Applied Science. And in parallel, I founded my company, IBI, that is a spin-off, a startup, is a biotech company focused on bone regeneration. So just have a look about the product that you can find on the market. So today, autografts still remain the gold standard in the market, so the bone that you harvest from the patient. Then you have cadaveric bone, that is the 80% of the US market. Then you can find also some xenograft material, especially bovine bone, equine bone, and porcine bone. And then you have alloplast material that normally they are quite cheap and they are quite far from the gold standard. So quite far from biomedical, biological, biomechanical, and chemical point of view. So here you can see some scanning electron microscopy of some product that you can find on the market. Starting from the right side, you can see a spongy bovine bone distributed by and produced from a Swiss company. Then you can see another spongy bovine bone. Then you can see a spongy cadaveric bone, and then a cortical bone. So if you see the spongy bovine bone, you can see that from a morphological point of view, these products are quite different. This is due for the cleaning treatment. The first one, they adopt cleaning treatment at very high temperature. So they change the chemistry of the osteopatite in order to reduce the volumetric reabsorption during the first days, the first week after the implantation. The other approach, the second product, they use a cleaning treatment with a chemical attack. So this to preserve the chemistry of hydroxyapatite, so to promote the remodeling, but obviously they have some problem in reducing volume immediately after surgery. So here we can see some papers, some result on papers. And here you can see that in the case of BIOS product, the first on the right side, the previous slide, you can see that if we perform an histological analysis, a biopsy, after 10 years, you can see that there is no remodeling. So you can see some bovine bone particles still in the body. So the new bone can grow around this particle, but there is not the possibility of remodeling, a complete remodeling. So our approach is quite different. So we decided to start from bovine bone from Australia because it's a BSE-free area, so no mad cow disease. And we decided to adopt bovine bone because we have a very track record of safety of the product, thanks to other companies that arrived on the market more than 50 years ago. So we decided to adopt a cleaning treatment at a low temperature, so it's a chemical attack, to remove all the biological substances inside the bone, so collagen, blood vessels, cells, fats, and everything. So we harvest just the hydroxyapatite of bovine bone, and then we adopt a reinforced treatment. So we put some biodegradable polymer in order to improve the mechanical performance and the volumetric maintenance. And then we put also some collagen fragments, some proteins, in order to improve the biological activity of the product. Here you can see the product from a morphological point of view. We can see that we try to mimic a non-ultra-compact cortical bone from morphological, chemical, but also mechanical point of view. Here we can see, if this, okay, that after this treatment, we still maintain an open and interconnected porosity. That is a key point for degeneration. So the blood have to arrive everywhere inside the graft, and obviously have to exchange nutrients and waste. Yeah, if you perform an apple cut of this bone graft, you can see that our treatment is a kind of coating that we put onto the surface of the trabeculum, the inner surface of the material. So here you can see in the red arrow, we can see phosphoric calcium. So we can see that is the xenograft material. While the blue arrow show the coating that we put onto the material. So it's a very, very thin coating. It's around 10 micron. And we know that from some analysis, but also from the literature, that after 4 months, this polymer is completely degraded by hydrolysis. So there is no strange reaction, degradation of this material. From mechanical point of view, we can see that in this case, we perform just some loading, and we can see that the material shows some mechanical performance higher, 4 times better than the best performing competitor. So the material, the behavior, the mechanical behavior of this material is very, very similar to a cortical, non-ultra compact, ill-thin human bone. As you can see from this video, the material is very, very easy to shape. You can cut, you can screw. There is no deformation of powder that obviously is quite noisy during the surgical approach. And the mechanical performance of this material permitted that you can fix properly the material to the site, avoid the unpredictable fracture. So you can obtain a very, very good primary stability. That is a good point to avoid deformation of fibrous tissue. So you can see that this material is very, very, very strong, easy to shape, easy to fix. So about the biological aspect, we put some gelatin, so material that contain the RGD sequence. In this case, the cells can connect immediately to the surface of the bone, especially osteoclasts that have to destroy the necrotic bone, the xenograft bone, can connect immediately to the surface of the graft. Here you can see some in vitro analysis. You can see that the cells are very well spread onto the surface of the bone. From histological information, in green you can see the bone, the cells, but you cannot see the polymer. So you can see that the cells can connect to the surface of the polymer. And then after the degradation, they start to create a layer onto the surface of the bone. Here we can see some biopsy, some histology in human, obviously, after 4 months. So the black arrow can show the empty lacunae, so you can see the xenograft material. And you can see that there is a new bone formed. You can see the woven bone. And you can see that we have some growth lines, so osteoclasts that are still creating bone. After 7 months, from another biopsy, you can see that there is a large osteoclast that is still remodeling the remaining part of the bovine bone. And after 2 years and a half, we can see that there is no more evidence of the graft. We have a very good vascularized new bone formed, so the integration was really complete. We obtain a very, very good bone. From this paper, from this study, we can see performing 10 patients. We can see that also from the in application like dental application or maxillofacial application, especially in this case in maxi sinus lift, we can see that after 7 months we can obtain mature bone with a remodeling of 60-70%. Obviously, before to arrive on the market in 2012, we performed all the analysis requested from the regulatory system, so cytotoxicity, sensitization, genotoxicity, and so on, and all the results of these tests were already published. Here we can see that another key point was the hydrophilicity of the material thanks to the protein added to the surface of the bone. And this is another key point because obviously the blood has to arrive everywhere to permit to the cells to arrive everywhere and exchange nutrients and waste. Thanks to the mechanical performance of the material, we decided also to invest in this service, if you want. So started to produce custom-made graft. So here we can see rendering for the regeneration of a very small area for dental application, but obviously there are a lot of software for planning, obviously different cases in different applications. Now I want to show you some clinical results. This was the starting situation. This case was performed in Sweden. And you can see that obviously, thanks to the custom made, you can create the right piece for every kind of defect. Here you can see after 2 months, we can see that we have to wait some months for a good regeneration. And here we can see that after 8 months, we have a complete regenerate of the bone. There is no more border, and we maintain also a perfect volumetric shape of the graft. Here you can see the reopening after 8 months. The surgeon decided to place 7 implants, and here you can see the result after just 1 year. We perform also some more complicated cases in maxillofacial application. So this was the starting situation. So we have to replace all the zygomatic area and the orbital floor. Here you can see the 4 pieces produced. Some phase of the surgery. Here you can see the previous situation and after the surgery. Here you can see the case after 9 months with a very good integration, volumetric maintenance, and after 2 years and a half, he decided to remove the scar, and we decided to take a biopsy of the bone just to evaluate the integration. From histological point of view, you can see that also in this case, there is no more evidence of the xenograft. We can see a very, very good vascularization, very good integration. This is another interesting case performed in Naples. This is an anesthetic surgery after an implant cancer removal. Here you can see that in this case, we perform a kind of puzzle technique to reconstruct all the area. You can see the follow-up after 10 months. And after 2 years, we can see that the integration, the volumetric maintenance is really good. We are active also in different applications. So in vertebral replacement, cancer, in trauma, in common orthopedic surgery. Here we can see some very, very common surgery. In this case, it's quite interesting because after removal of the cyst, normally they have to put a prosthesis, titanium prosthesis with a lot of fixation. But normally, as in this case, when the patient is quite young, it's not so nice to put some titanium implant because after some years you have to reopen. So new surgery to remove the implant or change the implant. In this case, the surgeon decided to adopt a new technique without a fixation device. So you can avoid a second surgery to remove the titanium implant. This is another interesting project, the osteotomy. Here you can see the normal approach performed with this material. And we are working with a collaboration with an hospital in Turin also to try to avoid the titanium fixation, so to avoid the second surgery. Yeah, another interesting application. So this is a cyst removal, and you can see that after 6 months, there is no more evidence of the graft and a perfect integration. This was the first complete vertebra reconstruction in the world with a biological material. Now we perform 5 cases, and we are performing a study in collaboration with IOR in Bologna. And now we are moving, we are trying to find a new solution, standard solution, adopting a kind of Lego technique to give to the surgeon standard pieces to replace vertebra in case of cancer. So this is the situation after the surgery. This is another interesting case because also in this case, in the iliac aneurysma cyst, normally they have to put a lot of titanium plate and screws. In this case, they achieved the result without titanium devices, and here we can see the result after 2 years. So here you can see the traditional approach and the approach with this innovative material. Obviously, sometimes we have to use also the titanium plate, as in this particular case where we reconstruct a complete hip after tumor removal. In this case, we are in collaborating with some company that can produce a customized titanium mesh with a trabecular structure in order to reduce the use of titanium. This is the last case that I want to show you. This is a compassionate case. This is a child, 8 years old. 3 years ago, the surgeon decided to go with the amputation of the leg. And we try to give a chance to this guy, to this child. So they removed the osteosarcoma. We try to replace the bone with this graft. So here you can see some phase, the radiography immediately after surgery, after 2 months, where we can see a very good integration. Here we can see the situation after 2 and 6 months. And after 3 years, we can see that the guy is safe with his legs. So this is our numbers. So now we have active in more than 30 countries. We have a staff now with 20 people. We have 3 trademarks and more than 50 scientific publications. So thank you for your attention.
Thank you very much, Gianni, for this amazing presentation. So I saw a lot of applications that you have found for your material. So it's really impressive how you can manage different bone pathologies and bone sites. So I always ask if there is a question, if you want to think to a question in a while. I want to ask about the last case with the osteosarcoma. So they have a very big defect. So how the bone could migrate from from one side for such a long way for like 20 centimeters or whatever. So this is really impressive. So
yeah, this was I think the largest case that we performed. And in this case, the surgeon decided to harvest the fibula, one of the fibula to the child, and create a kind of sandwich in order to promote a faster integration.
So you had like a side.
So graft fibula graft in order to, and also we have to think that in most of these cases they have also some radiotherapy and everything that reduce the viability of the cell and everything. But in this case, we obviously was one of the main case, but we were quite lucky, and the integration went properly. Yeah. Obviously, in future with stem cell technology and everything, probably we will find new solution to can replace bigger pieces.
Yeah, and since the child grow pretty fast, so you had this integration. So as fast as the growth of the bones.
Obviously the two legs can go at the same time. So, but obviously now he has two legs instead of one. So
sure.
The result is good.
Yes, fantastic. Thank you, Gianni. So, is there any question? So, if not, I think that we